This recording begins another topic in our study of human anatomy and physiology. In chapters 6 and 8, we studied the bony skeleton and then the various joints that connect the bones of the skeleton. And now we're entering into chapter 9, in which we'll learn about the muscles that produce the movements of the skeleton around those joints. So here's a little sort of guide to the topics, the subtopics we'll be looking at. We'll start out looking at some anatomy as always so that we'll know what is there, what kind of materials we're dealing with, and then we can talk about the physiology. Here's a little diagram to begin with macro, the largest scale anatomy of a muscle organ, and then we'll work our way down to some more incisive looks at the microscopic anatomy. So the muscle organs are all attached to the skeleton by tendons, and we see this dense regular connective tissue attachment to the bone, and that tendon is continuous with a connective tissue jacket around the muscle organ called the epimysium. There's all collagen fibers there that are surrounding this muscle. If we look inside this cross section, we see a bunch of kind of white halos, and those halos represent connective tissue boundaries, wrappings of a subunit of the muscle called a fascicle. A fascicle is a bundle of muscle cells, or muscle fibers they're called. And so the connective tissue wrapping around the fascicles is called the perimysium. And that perimysium is in addition continuous with the tendon also. Each muscle cell that we see within the fascicle is surrounded by a fine connective tissue covering of its own called the endomysium. All these connective tissue coverings are continuous with the tendon. So the tendon firmly anchors every part of the muscles down to the cellular level, right to the bone. And in addition, the cytoskeleton is what we're looking at inside this muscle cell, is anchored to the endomysium. So we have a strong connection to the bone. Here's just a photograph of a cross section through some muscle to illustrate those same structures. Here's a picture of a micrograph of a couple of muscle fibers, and we can see that muscle fibers, as we've talked about before, skeletal muscle fibers have striations or stripes. We're going to take a look and figure out what is it about the arrangement of the cytoskeleton proteins that produces that effect. So here's again looking at our muscle fiber, the membrane, the sarcolemma, which is just the fancy way of saying plasmalemma. Sarco just means of muscle. The myofibrils are the organelles of these muscle cells, and they're not just organelles, they represent the cytoskeletal arrangement. The microfilaments in the cytoskeleton are what the myofibrils are comprised of. But the orderly arrangement, you can see these repeating structures or arrangements of microfilaments in a myofibril, and then every myofibril is exactly lined up in such a way that wherever there's a dense composition of protein, it forms kind of a dark band as you look through the entire muscle cell, and wherever there's a less dense region of protein, it makes a lighter band. So it's so intricately organized that we can see right through the lighter areas, and it forms those striations. Here's a cartoon version of a myofibril, and we can see the two microfilaments we're talking about here are the myosin filaments shown in red, so-called myosin thick filaments because they are physically thicker and heavier in dimension, and the actin proteins, which are interdigitated with those myosin filaments shown in blue. And again, the arrangement of the filaments in these repeating patterns produces both the striations that we see as well as the functional unit of a muscle cell. Here's the functional unit from a Z disc to a Z disc shown in two-dimensional cartoon drawing to sort of really highlight what we're looking at a little bit more. The actin filaments are here in blue, attached to a plate called the Z discs. A bunch of structural proteins are there, and in between all the actin filaments are myosin thick filaments that have all these bristly looking so-called globular heads there. And this unit from Z disc to Z disc, this the sarcomere is the smallest functional unit. It can physically shorten, and that's what happens when a muscle contracts. All the sarcomeres that make up all the myofibrils shorten, and that sums up to produce a significant reduction in the length of a muscle, the contraction of a muscle. This is actually a cylindrical structure, of course, and if we look at it in cross-section, we'll see some interesting observations. If we cut through the sarcomere where the protein is densest, we see the actin and myosin filaments are overlapping. Here's what it would look like in cross-section, and you can see that every myosin filament is surrounded by actin microfilaments. Or if you look at it the other way, every actin microfilament is surrounded by myosins. So they're right in context of one another, and that'll prove to be very important in our discovery of how muscles contract, how the sarcomere shortens. On the other hand, if we look at a place where there's just actin filaments, and maybe some elastic proteins in here drawn in yellow, we'll see that it's a much lighter situation, and that's what produces the so-called I bands, the light bands of the striations. The dark bands are produced by the places, the A bands, the regions of the A bands where there's myosin and actin in a very dense packing of protein indeed. It just so happens that the bristles of the myosin heads, the myosin filaments, the so-called globular heads, are the working units that actually move the Z discs closer together by tugging on the actin filaments, and so that this only happens in the places where the two types of filaments overlap and there are no myosin heads in the middle section here between the actin filaments and the sarcomere. So here's a closer look at the myosin thick filaments. A myosin thick filament is made up of a couple of hundred subunits that take this form: a pair of myosin proteins with the tail regions, the linear kind of tail regions, twisted into a strand, and then globular heads that are attached by a hinge to the strands, and then the whole thing assembled into a polymer, essentially, with all these globular heads sticking out like bristles of a cylindrical hairbrush. The actin microfilaments are made up of many so-called globular actin or G-actin subunits, and they're assembled into long strands or threads called filamentous actin or F-actin, and then a couple of F-actin filaments are twisted together to produce the thin filaments, the thin microfilaments. The little dark spots we see here are myosin binding sites. The myosins, the little dark spot drawn on the globular heads of the myosins, is meant to represent a place where myosins can bind to actin subunits with high affinity, high grip. So if you let a myosin head get in the vicinity of one of these actin units, it'll stick right on. Well, when the muscle is at rest, it can't stick on because the actin filaments are completely wrapped up in a sheath of tropomyosin. These orange threads are called tropomyosin, another protein called a regulatory protein, because it's going to block the interaction of actin and myosin and prevent the muscle from contracting, the muscle cell from contracting. We also see another regulatory protein here called troponin, and troponin is like a handle attached to the tropomyosin, and we can move it. And if the handle, the troponin, changes conformation, then it'll move the tropomyosin out of the way, and those binding sites for actin will become revealed, and we'll have something dramatic and exciting happening. So let's wait for that. Here's a beautiful micrograph showing the actual physical nature of these things. The thick myosin filaments are shown here, and here we can see the thin actin filaments, and we see some what are called cross bridges between the two. In this case, the myosin heads have been allowed to attach to the actin filaments, and we have cross bridges. And that's the sort of beginnings, the prerequisite for muscle contraction is the formation of those cross bridges. So that's pretty cool. In order to understand the regulation of muscle contraction, we need to introduce a couple of more features of a muscle cell, and this is the whole muscle cell again with multiple myofibrils in there. What's shown in blue, where we see these sort of chunky storage tanks or reservoirs on the ends of a labyrinthine connection of tubules is the endoplasmic reticulum. All cells have endoplasmic reticulum. It's just that in a muscle cell, it goes by the name sarcoplasmic reticulum because, again, muscle cells have been studied for so long by so many and with the investment of so much collateral that everything has its own special name, and so we call it the sarcoplasmic reticulum, the SR. Well, the sarcoplasmic reticulum has the special property that it's the location of the storage of calcium ions. All the cells in your body, when they're at rest and not doing anything dramatic, have no calcium in their cytoplasm. I wrote down a value for the amount of the concentration of calcium in a resting muscle cell. It's approximately 10 to the minus 7th molar. Molarity is a measure of concentration, as you may know. When you have a power with a negative sign in front of it, that means that's the same thing as saying 1 divided by 10 to the 7th power molar. In other words, 1/10,000,000th molar calcium is found in the cytoplasm. It's nada. You can just call it 0 at rest. But there's a lot of calcium stored inside the sarcoplasmic reticulum. That's our reservoir of calcium, and as we'll see, that'll be an important property of the muscle cell when it comes to turning on contraction. Here's another interesting feature. The cell membrane, the sarcolemma, has tubular processes or invaginations that cross through the entire muscle cell. If we could somehow see down through or climb down into this pipe-like construction of the membrane, we could crawl all the way through the entire muscle cell and come back out the other side and never cross the cell membrane. So the cell membrane penetrates way down into the muscle cell, and in fact these transverse tubules or T-tubules of cell membrane snuggle right up against these kind of bulky end caps of the sarcoplasmic reticulum. They're called terminal cisterns. A cistern is just a reference to a time before indoor plumbing in homes, in which the rainwater was captured from the roof and piped down into a tank, a storage reservoir in the cellar of all that rainwater, and that became the water to use for cooking and washing and so forth, because there was no running water. And so the cistern was an important part of daily life, especially in rural areas, and you just lift up a trapdoor in the floor of the kitchen and dip down with a bucket to get some water. So that's why the anatomists way back in the day named these cisterns. They're the tanks, or the big voluminous storage areas of the sarcoplasmic reticulum. So we have transverse tubules of the cell membrane that penetrate all through the muscle cell and are in direct contact with the terminal cisterns of the sarcoplasmic reticulum, where all the calcium is squirreled away. Well, it was many years of intensive research before anyone could figure out exactly how mechanically muscle cells can shorten. What makes them physically contract? There's a lot of biochemistry known, but not too the mystery of how muscle cells contract took a long time to discover. Well, one of the observations that led to the breakthrough was looking at a micrograph of a muscle that was stretched to its fullest length before being fixed and stained. And what we see here is the sarcomere from Z disc to Z disc, and we can see the dimensions of the A band, the dark band, and the dimensions of the I band. The I bands are very broad, and the A bands are, as we see here, in dark. Another preparation was made of a muscle that had already been contracted maximally and then fixed and stained. And the A band has exactly the same dimensions, but the I band has been reduced drastically to a much narrower dimension. And in fact, the Z discs are much closer together. And it became, an epiphany occurred when someone realized that what was happening is the actin filaments were being pulled towards the center of the sarcomere. The Z discs are being pulled closer together during the process of muscle contraction. The microfilaments are sliding along one another and doing work. So as we're about to embark on a discussion of how muscle cells can respond to stimulation and undergo contraction, we need to introduce a couple of membrane proteins called channels. They're transmembrane proteins through which ions can diffuse. And there's two types of channels in the muscle cell membrane that can open and close. They're switchable or gated, we say. There's chemically gated ion channels, such that when a neurotransmitter binds to the channel, it's a receptor, and it's also a channel. The channel opens and ions may pass through the membrane that were otherwise trapped outside or inside. Voltage-gated ion channels are closed when the membrane is at rest, but then something is going to happen by way of a change in electrical properties of the membrane, and it's the electrical properties that control the voltage-gated channels. Those channels will then pop open and ions will be allowed to cross the membrane. So let's take a look at a couple of cartoon illustrations. Here's an acetylcholine receptor in the muscle cell membrane, and some acetylcholine, the neurotransmitter of skeletal muscles, has been released and has bound to the channel and has now opened. Without the acetylcholine, it's closed, and when the acetylcholine comes on the scene, it opens up and now ions can pass. Electrogenic channels, or electrically gated channels, are closed when the membrane is at rest, but in this case we're looking at a membrane that has been already activated and has been altered electrically, and we can see a couple of little hinged trap doors that if we pull those little trap doors downward, you can imagine the trap door closing in the middle and shutting off that channel, but now we've lifted up those trap doors or those levers and opened up the channel because of some electrical change in the membrane. So those are two types of channels we're going to be discussing as we find out what are the electrical properties, the unique electrical properties of both neuron membranes and muscle cell membranes. So join me next time to learn about resting membrane potentials and action potentials.